Hello, let us have a very brief but very interesting discussion on southern blotting. Imagine you want to identify a specific DNA fragment from a set of DNA samples. You can do that by a molecular biology technique called southern blotting. What is to be done? This is the sample. Now to the sample, you are adding a restriction endonuclease so that this DNA will be cut into fragments. Right. Now you will be putting this particular DNA into alkaline solution for denaturing it, for separating the double bonds between the DNA strands. Okay. Now this will be transferred into an agarose gel for an electrophoresis process, which will be separating the DNA fragments by size. So smaller fragments will be moving towards the positive end very faster. Higher will be a bit slow. Now what you will do? You will be now do a process called southern blotting. In this process, what will happen? The DNA that is separated by size, that will be this entire gel will be transferred to a nitrocellulose membrane or a nylon filter. So after doing this blotting process, it will look like this. Now within this somewhere there is the specific DNA fragment of interest. For that, what we will do? You will incubate this nitrocellulose membrane with a radioactive or fluorescent labeled probe. Okay. Now this probe is a short single stranded DNA which is complementary to the target DNA of our interest. Now after the excess probe is washed off, you can see that only one place will be emitting the fluorescence. Okay, that will be the specific DNA of our interest. This will be seen clearly when it is exposed to an X-ray film. Okay, this simple thing is the southern blotting. Now I'll give you some notes regarding this thing if you want. So you can pause the video and take the steps. Yeah, for detecting specific DNA sequences in a DNA sample. Now, what are the and it is created by Edwin Southern. That's why it's known as southern blotting in 1970s. Right. So what are the steps? First, you will be fragmenting the DNA using restriction enzymes, restriction endonucleases. This enzyme is having a feature that it will be recognizing a specific DNA sequences and cleaving that particular DNA at that specific sequences. Then electrophoresis means separating by size. Then that will be transferred to a nitrocellulose or nylon membrane. This process called blotting. Then we will be incubating this with a radioactive or fluorescent labeled probe. That process is through that process hybridization of the probe with the target DNA happens. Then that can be detected using an X-ray radiography. So that bound probe, that particular segment indicates the presence and size of the target DNA sequences. You can measure, okay, it is attached to this particular site and our target DNA is this much long, kind of thing. So what are the applications? First of all, gene identification. Okay, within a complex DNA sample, you want to identify a specific gene that is possible. So then the studying of the DNA structure or gene structure, organization, etc., ATCG pairs, etc. Then if in one particular DNA sample or a gene there is a point mutation or insertion, deletion or rearrangement, whatever, that can be done. In forensic sciences, crime scenes, identification of suspects or victims through DNA fingerprinting. Yeah, DNA fingerprinting, that means in the southern blotting we are identifying a specific DNA of interest, right? So definitely it's an application of DNA fingerprinting. Paternity testing by comparing DNA sequences between individuals. Okay, it is because it's having the ability to detect specific DNA sequences. So just do the blotting process and incubation with the probe. The child's DNA can be made the probe, so that will be attaching to the father's DNA. Then that will be then disease diagnosis, identification of genetic disorders, mutations, genetic abnormalities, etc. DNA fingerprinting. Okay, within a population, you are measuring the DNA. You are sequencing the genome of a particular breed of sheep, for example. then you may at times get a unique genetic profile, so that particular thing may be bred continuously for increasing its population, etc. Right. Then comparison of genetic material from different species. You are comparing uh, a bat and a human being. So you can see that the bat's like palm and the human palm is having some evolutionary uh, connection. So you can compare the genome. And now you are importing one particular product. You can identify whether there is a genetically modified organism or transgene present in this particular thing. Then you can study how the viral DNA integrates into host genome, etc. So these kind of things can be the applications of southern blotting. I hope it's very clear to you. So we'll be winding up. Thank you.